All right. Hi. No one's lost their child. I am the speaker. My name is Michelle Meas, and I'm a student and researcher at the University of Illinois at Urbana-Champaign in the College of Microbiology. And this is Bioweaponry 101, how to theoretically kill a continent. Fun fact, the theoretically didn't used to be there. They emailed me when they accepted my paper, and they were like, a lot of people on the board were spooked. Can you put theoretically in there so people don't think you're actually trying to kill an entire continent? Well, I suppose that's fair, I guess. Quick disclaimer, this is not at all a talk that's against the use of genetic manipulation to create new organisms or even to manipulate bacterial genomes. There's a lot of cool stuff in like nutrition and medicine that have been doing work that's saving lives. There's a polio virus that's been sort of genetically engineered to target glioblastomas in the brain that is showing some promising results right now. So this is actually a tool that's been used for a lot of good things. It's just sort of a double-edged sword. The more we learn about it, the more potential it has for good, and the more potential it has for bad things, like bioweaponry. So, brief history of bioweaponry. The actual advent of bioweaponry is arguably somewhere around the 14th century BC. But it's sort of hard to trace back what is a deliberate use of bioweaponry versus a sort of accidental course of natural epidemics. And so nothing really gets interesting until these two men show up. So this is Robert Koch and Louis Pasteur. If medicine and microbiology was a religion, these two would be fighting to the death over who gets to be Jesus. They basically founded the entire theory of germ theory of disease. So basically, the idea that disease doesn't spontaneously arise from within a person, it's actually particulate. So there's little tiny particles that cause disease. And this is important because they also sort of proved that you can distill and culture these sort of pathogenic molecules. And you can, if you can sort of have these and breed them, you can then stockpile them and create weapons. And this was in the late 1800s, and you actually see the first sort of instances of people researching biological weapons as we know them in around the World War II era, so early 1900s. But things don't get super interesting until somewhere around World War II. So after the Germans used a whole bunch of like chemical warfare, like mustard gas, chlorine gas, etc., in World War I, the Japanese were inspired by this, and they sort of started this research facility, or a network of research facilities rather, called Unit 731. It's actually known as the Asian Auschwitz. And there were a whole bunch of really awful sort of experiments done here, including frostbite experiments and children being like dissected for like seeing how their mother's STDs would present themselves in a child born from them. But one of the main goals for Unit 731 was the creation and the weaponization of pathogens that could, sort of ravage entire countrysides without having to lift a finger. The Japanese actually used a bunch of these in World War II when they were going into China. So they would do things like spray plague-infected fleas over the countryside or drop bombs with porcelain shells that had like disease molecules inside of them. A a very important pathogen that they used was Yersinia pestis, which is the bacteria that causes the Black Death or the bubonic plague. So they did this for over a decade, for about 13 years, and they didn't stop until the war was won, basically. So what happened, and why didn't we hear about this? Well, when Unit 731 was captured, Lieutenant Colonel Murray Sanders, who was a microbiologist, got a report of their sort of goings-on in Unit 731, and he reported it to General MacArthur. And the United States actually granted amnesty to a lot of these physicians in order to procure the data that they had sort of accumulated over time during the course of Unit 731. They not only wanted to keep this data, they wanted to keep this data as the only Allied force member to have this data. So they really didn't want the Russians to have it in particular. So actually in the mid-70s, late 70s, there was a bunch of no one really knew that this happened, and there was a bunch of cover-up by the United States government. For example, when the Russians tried some of the physicians that were captured from Unit 731, the United States covered up a lot of these trials because they didn't want people to sort of know that this happened, because if people knew that it happened, they would sort of realize that if we were in charge of the trial of the other Unit 731 physicians, then we didn't prosecute them. That could be like a really bad thing for the United States government. That would be some really bad PR. So the United States biological weapons program didn't really start until 1942, but it didn't take off until after the procurement of this data from Unit 731. And, you know, the United States, we like to think of ourselves as a lot better than all of these other people who experiment on, you know, unknowing civilians and prisoners of war, but actually from the time between the 40s and the late 60s, the United States did a whole bunch of tests on its own citizens to simulate biological weapons attacks and to sort of test the dispersal methods that they created. One of the most notable ones was the infection of the San Francisco Bay Area in the 50s with Serratia marcescens. So this bacteria, we thought it was very non-virulent, very safe for people, and we actually put it in aerosolized forms, and we had a ship that would go up and down the United States coast and spew tons and tons of, you know, aerosolized pathogens into the air, and people would inhale it. People would inhale like millions and millions of colonies over the course of days and weeks that this took place. And after all of this started happening, we saw a bunch of croppings up of infections of this bacteria in particular. And before this, there had been no recorded cases of a S. marcescens infection in this area. And there were a known 11 cases of urinary tract infections as a result of this bacteria, and a whole bunch of other cases of pneumonia that can't technically be linked to this because pneumonia is not caused by any one bacteria, but it was a dramatic increase from past years. And the United States government maintains to this day that this is totally coincidental, and nothing that we did had any effect on, you know, the population. But the data sort of speaks for itself. So during the course of the United States Biological Weapons Program, we weaponized and stockpiled 7 agents. You'll recognize some of these as, Bacillus anthracis, that's anthrax, Francisella tularensis, which is tularemia or rabbit fever. There's a bunch more, but we definitely researched more. There was more research being done than just this 7, and there is potentially more weapons production made from these 7, but this is what we know. It's actually been theorized that over the course of the Cold War, the United States and the Soviet Union stockpiled enough biological weapons agents to kill literally everybody living in the world. And so, in 1972, the World Health Organization got everyone together and sort of had them sign this agreement, which had 3 parts. They prohibited the possession of bioweaponry, except for quote-unquote prophylactic, protective, and peaceful purposes. I don't know what peaceful purposes you would have for bioweaponry, but it's in there. They also, prohibited the development of bioweaponry and dispersal systems for offensive purposes, and they called for the destruction of the existing properties that contained the stockpiles. So this came into effect in 1975, but it obviously had some glaring shortcomings, such as what constitutes as a peaceful purpose for having bioweaponry. What is an offensive versus a defensive dispersal system for bioweapons? And largely, the sort of research into bioweaponry continued into the modern day. There's actually cases of the Clinton and Bush administration having talked about bioweaponry and being investigated for sort of violating this convention, but obviously it was not very conclusive. So some modern cases of bioweaponry and bioterrorism attacks include the 1984 Rajneeshi cult, which was sort of a cult located in Oregon. And what they did was they took the bacteria that causes salmonella and they infected a bunch of salad bars across the state. And they did this to dissuade or to keep people home from voting so that theoretically their own candidate would be able to win the election and hold office. There were over 700 cases of infections and around 40 or 50 hospitalizations, but nobody died because of it, which is good. In contrast, the anthrax letters was sort of a thing that shocked the nation. It happened on September 18th, 2001, and the letters were sent to, two were sent to United States senators, and 2 were sent to sort of media outlets. And these letters were dated as September 11th, 2001. And only 4 letters that we know of were sent. However, 17 people contracted infections and 5 of them died. This sort of shows the lethality of Bacillus anthracis, the bacteria that causes anthrax. Despite the fact that only 4 letters were sent, 17 people got infected because this bacteria is so virulent and so pathogenic. We actually don't conclusively know who sent these letters. We had a suspect who worked at one of the United States bioweaponry dispersal systems and laboratories back in the day. But he actually committed suicide while under custody. So we actually don't know where these came from. So really quick, some basic terms that I'm going to be referencing throughout the presentation. The ID50 is the dose for 50% of people to have an infection. So basically, it's going to be measured for bacteria, CFU, or colony-forming units. So a colony-forming unit is basically just a single cell that is viable and proliferates throughout your system. The term virulent, when I say that, it means really severe or infectious and harmful, especially to humans. And a virulence factor is a molecule that's produced by a pathogen that allows them to become virulent. So it allows them to, colonize the host or hurt the host or get out of the host. An antibody, you probably already know what an antibody is, but it's a protein created by the immune system after identification of a pathogen. So an antibody can bind to certain things on a pathogen, and it can incapacitate or precipitate it and make it clump together and come out of the body, or it can mark the sort of pathogen for destruction by the immune system. And an antigen is just the protein on the surface of the pathogen or inside the pathogen that the antibody binds to. There's a difference between infectious and contagious. So infectious diseases are diseases that can, cause an infection, cause a sickness in a person, whereas contagious is sort of a communicable illness from person to person. So not all infectious diseases are contagious, and not all contagious things are necessarily infectious. And also, lastly, the term zoonotic just means a disease that's normally in animals but can cause disease in humans. So what do we have as protective measures for bioweaponry? Not that much, actually. So after the anthrax letters of 2001, we sort of started this program called the BioWatch program. And what it did was it sort of repurposed EPA filters around the nation so that they could then be sent to labs. And the labs could then look at the DNA of the stuff that's trapped in there and compare it to a database and see if anything's sort of pathogenic. It was revamped in 2003, and it was distributed to around 30 cities around the country, and it was made more efficient. But there are some drawbacks. For example, there's not a lot of reliability in this system because it often gives false positives. And this is because a lot of the things that we believe can be weaponized and created and turned into bioweapons are actually pathogens that we can find in everyday life. And cousins of these pathogens can also be found just in the air as non-virulent organisms. And so there's been over 50 sort of false positives from the time between 2003 and 2008. And none of them have been true, obviously, because we would have heard about it. But this is sort of like an oversight on the part of, the critics, because when you screen for the presence of bioweapons in the air, it's sort of like a smoke detector. It's going to go off if you burn a piece of toast, but you should still check it if it starts beeping. Because if you decide to not check it when it starts beeping for a burned toast, the burned toast can catch on fire. And then your house can burn down, and that would be, pretty bad. So there was this huge movement to revamp the BioWatch program somewhere around 2008. And so what that would do would be to create sort of a lab in a box, to sort of eliminate the middleman so that the filter could check the DNA, capture the pathogens, check the DNA sequences, and then run this like data through a computer database system without having to go through a lab. But that would cost a whole bunch of money, and no one wants to pay for it in the Senate because they don't really have a lot of faith in it. So we're still going off of technology from 2003, and that's supposed to protect us. But in reality, this is probably not what's going to protect us because it's old and it's been fallen into sort of disuse. So what we're really relying on is the ability of physicians to diagnose diseases accurately and to report things that are suspicious. So that's not super great because the state of medicine in the United States is such that we have a physician deficit, and we have an overwhelming number of patients for the number of physicians we have. So when you go to the doctor and you think you might have a flu and you have flu-like symptoms, the doctor probably won't test you for flu. He'll probably just be like, yeah, your symptoms point to flu, and here's some Tamiflu if you want it, and go home, get a lot of rest, and drink fluids and all this stuff. But that would be a problem if we had a pathogen attack that emulated a lot of flu symptoms in its early stages, because we wouldn't catch it fast enough. And that would be incredibly bad. So let's just take a break. This is a bio talk, so here's a bunny. This is an eastern cottontail. It's indigenous to most places in the United States, and it's just cute. I like him. He's got a nice look in his eye, but they are not very harmless to humans, or are they? Actually, they're really not. They're really not harmless. They're not harmful at all, but they do carry Francisella tularensis. They are the most well-known vector for this bacteria, which causes tularemia, also known as rabbit fever. So there are 4 subspecies of this bacteria in varying levels of virulence, but we'll focus on the subspecies tularensis, which is the subspecies that is present throughout North America, and also the subspecies that is most likely to be weaponized. So Francisella tularensis is a tier 1 bioagent labeled by the CDC, which means it has a high potentiality to be turned into bioweapons. And actually, the US did that prior to 1975, before they allegedly destroyed all their stockpiles. And they aerosolized this, and they, were intending to stockpile it for use against other people. So, yeah. So how does Francisella tularensis get to you? So we actually don't know what the original host for this bacteria is. There's a lot of evidence that it could be a protozoa, for example, an amoeba, but we actually don't know where it exists normally, because it doesn't survive forever in the environment and in lakes, and it kills rabbits and rodents, and it doesn't really proliferate inside ticks. So the general sort of method of a human getting this would be, we don't know the original source, and then a rabbit gets that, maybe from like drinking it up or something, drinking up the original host through infected water, etc. And then this rabbit can get bitten by a flea or a tick, which can then transmit it to you. Alternatively, this rabbit can be run over by a lawnmower, and the sort of infected particles from this rabbit can go into the air or into the water, and you can get it from breathing that air or drinking that water. And actually, Francisella tularensis infections across the United States is mostly like centered in rural areas, where they would be like going through with huge combine harvesters, and they might run over a rabbit, and the dust gets in the air and they inhale it. So this is actually not that uncommon of a pathogen. So the symptoms of a Francisella tularensis infection would be that you'd have like 1 to 14-day incubation where you might not have symptoms, but the average is around 3 to 5. Then you start having coughs and difficulty breathing and fever and chest pain and all this other stuff. And eventually you can get to pneumonia or sepsis, which causes septic shock. This subspecies of Francisella tularensis actually has an incredibly high mortality rate. If you leave it untreated, it actually can go from 30 to 60% mortality rate. So you can die 30 to 60% of the time if you contract this disease and you don't go to a doctor and you don't get treated, or you don't get treated properly. So how does this pathogen work? Well, first we're going to have to take a step back, and we're going to have to talk about the immune system. So in your immune system, there are these sort of white blood cells or lymphocytes called macrophages. And you can see what the macrophage is doing up here. It's that like little molecule over there is a bacteria, and it's sort of running away. It has these little like cute helicopter propellers that sort of like fling it around, and it's essentially just eating that bacteria, and it acidifies it, and it breaks down the bacteria. And that's actually how your body avoids a whole bunch of diseases, because you have these all over your body. They're one of the first lines of defense for your immune system. And they carry an incredible amount of weight in your immune system. First of all, they destroy bacteria. And second of all, if the infection gets too out of control, they will take this bacteria and break it down and take the broken down pieces and present them in your immune system to T cells and B cells, which are things that create antibodies and create a natural immunity to things. And so the macrophage is one of the most critical components of your immune system. What Francisella tularensis does, instead of being engulfed by the macrophages and being acidified, is it sort of breaks out of this sort of protective coating that the macrophage puts around it before it acidifies it. And it gets into the macrophage, and it proliferates and sort of lives in the macrophage for a while. And then after it's, divided enough times and multiplied and grown stronger, it forces the cell to commit apoptosis. And apoptosis is programmed cell death. And this is how the bacteria then releases hundreds of thousands of copies of itself into your body. So how does it kill you then? Well, other than the decimation to your immune system, which can lead to severe secondary infections and often cause pneumonia, Francisella tularensis can cause sepsis and septic shock. So what septic shock is when the pathogen gets into your system. It can then go into the bloodstream. And your body sort of has a natural way of getting rid of pathogens, and what it does is it sends these macrophages and other immune system cells throughout the bloodstream. And when it senses a pathogen, it sort of opens the blood vessel walls so that these white blood cells can get out and fight the pathogen in the tissue. Because most likely, the pathogen is not in the bloodstream, it's in the tissue. But Francisella tularensis has this nasty habit of getting inside your bloodstream. And so when you open up these blood vessels, the blood just leaks out everywhere. Your blood circulates through your system once every 2 minutes. It's super fast. So everywhere in your body, the blood vessels are leaking. And you suddenly don't have enough blood to, cater to the needs of your vital organs and your brain. So you start to suffer organ failure. And if it goes for long enough, you can suffer suffer massive organ loss, and you can die from this. So there are treatments available, obviously. You would hear a lot more about this probably if there wasn't. We have antibiotics that can treat it. We have a vaccine that is partially effective against it because it's created from a different strain than this, most virulent strain. And one thing we do know about Francisella tularensis is that it doesn't have the capability to pass from person to person. So that's a good thing. So with that in mind, what is the viability of creating bioweapons? You essentially have to create a new organism. You have these super pathogenic organisms like Francisella tularensis, but a lot of them are not contagious because they're not evolved to be so. And a lot of the ones like Ebola that are contagious and do cause death so fast that they can't get outside of a very small population. You can't just make new genes, right? It's very, very hard to accurately predict how a gene is going to be translated into a protein and how it's going to fold, because there's going to be thousands or tens of thousands of sort of different factors that play into how proteins are folded and how they work and what they do in the body. So you can't just sort of de novo create this. We have antibiotics, we have modern medicine, we have vaccines. These are all things that have increased the lifespan of humans by decades. So why are we even worried about bioweaponry? So the problem with this is that you don't have to just create new pathogens. You can essentially let nature do the creating. And this is a short video clip from the Harvard School of Medicine, I believe, that is going to show you how that works.
So what we ended up building was basically a petri dish, except that it's 2 feet by 4 feet. And the way we set it up is that there are 9 bands, and at the base of each of these bands, we put a normal petri dish thick agar with different amounts of antibiotic. On the outside, there's no antibiotic. Just in from that, there's barely more than the E. coli can survive. Inside of that, there's 10 times as much, 100 times, and then finally the middle band has 1,000 times as much antibiotic. And then across the top of it, pour some thin agar that bacteria can move around in. The background is black because there's ink in it, and the bacteria appear as white. First, you see they spread in the area where there's no antibiotic, up until the point they can no longer survive. Then a mutant appears on the right. It's resistant to the antibiotic, it spreads, until it starts to compete with other mutants around it. When these mutants hit the next boundary, they too have to pause and develop new mutations to make it into 10 times as much antibiotic. And then you see the different mutants repeat this at 100. And after about 11 days, they finally make it into 1,000 times as much antibiotic as the wild type can survive. And so we can see by this process of accumulating successive mutations that bacteria, which are normally sensitive to an antibiotic, can evolve resistance to extremely high concentrations in a short period of time.
All right, so that's pretty scary. And some things that I want to point out is that every sort of branch along the pathway, that was a separate mutation from different strains of this bacteria. So let me see if I can get back to this image over here. So every node along this path is a new mutation that this one bacteria over here has, and each node is a different one. And so you can create not one antibiotic-resistant gene, but many antibiotic-resistant genes. And if you can create many antibiotic-resistant genes, it's very, very hard to create antibiotics that can combat that. And another thing that I want to point out is that in this paper, they showed that originally these mutants would grow a lot slower, but after a while they would start proliferating at the same rates as the original bacteria did. So this means that these mutants are normally less viable than their sort of normal, quote-unquote, cousins. But they can regain that viability very, very fast. And that's because they multiply at rates of hundreds per day. And we already see this in modern medicine today with CRE, which is an infection that is normally garnered in hospitals, and it's resistant to almost all antibiotics that we have on the market today. And if you contract this disease, even with all of the wonders of modern medicine, you have a 40 to 50% chance of death. Obviously, this varies on like your demographic and your age range, especially, but it's pretty scary. And we also have, C. diff. Everybody's heard of that, which is another mostly garnered in hospital and healthcare type settings, and it causes mortality in around 6 to 30% of the people who contract it. And this is a large range, right? But it mostly depends on your demographic. Like I said before, for example, C. diff often targets people with weakened immune systems and people who have just got off antibiotics, because this bacteria is a bacteria that just naturally lives in your gut. And the problem is normally that too much good bacteria, quote unquote, is killed off, normally by a round of antibiotics, sometimes by certain things that you eat, and then this is able to proliferate in your system. And so it's already inside of you. And yeah, so we may have already killed a continent if we let this get out of hand. Thankfully, there's a lot of research going on with antibiotic-resistant bacteria. A lot of it has to do with immunological responses, but there's a lot of very exciting research being done there. So if we can eventually find a cure for this antibiotic resistance that we're facing, what do we have to fear? Well, you can also create old viruses that are supposedly dead. And you can take these old viruses that we have no information of and no vaccines for and no sort of treatments for, and you can sort of raise them from the grave. So this paper in 2005 by Tumpi et al. Sort of demonstrated this in practice. So what happened is, in 1918, there was a Spanish flu epidemic, the largest pandemic in known history, and over 50 million people worldwide died of this flu virus. It was mostly thought to be because of, the World War I winding down, and there having been a lot of soldiers and a lot of close spaces and a lot of exchange going on the battlefield. And then all of these soldiers went home and dispersed the sickness that they garnered overseas to their families. And so what happened was they found some poor sap who died of this in Alaska and then got frozen in Alaskan permafrost, which is sort of like the microbiologist equivalent of striking gold. And so they could find intact RNA sequences from this virus. And they couldn't find all of it because obviously it's been degrading in the cold for this time. It's been sort of stopped by the fact that it's been frozen, but it's been degrading nonetheless. So they took all of these little snippets of the RNA sequence of this virus, and they reconverted it into DNA, and then they sequenced that DNA. And they ran all of these little sequences through a supercomputer and had it find all of the potential overlaps and then piece it together based on what the information they had. And when they then synthesized that one long strand of DNA and then converted it into RNA and injected it into the cells of mice, these mice cells then started creating this old virus, and the mice died because it's actually not just because of World War I that this was a really bad pandemic. It's because this is an incredibly virulent strain of the flu that it sort of sheds and contaminates 50% more than a sort of analogous flu virus that we have today. It's been described as the most avian type of human infecting flu. And that was pretty scary. But, you sort of have to have this weird circumstance of preserved RNA from viruses. So that doesn't actually happen that often. However, you can just create viruses on your own, because there are companies that will sell you specific sequences of DNA called oligonucleotides. And these oligonucleotides are, used by a range of microbiology labs across the nation. Like, they're very common. And so in 2002, a bunch of scientists got together and ordered a bunch of parts from these oligonucleotide synthesizer companies, and they glued them all together, and they created a polio virus. And you can just do that, apparently, which is scary. So you don't even have to have access to sort of like stockpiled weaponry to create these things. You just need a lot of time and money. But the polio virus is actually a pretty simple virus, and its genome is rather small. So at the time we were like, yeah, but it'll never happen to something really large and dangerous like smallpox. But it did in recent years. So what happened was this team of researchers led by I think Robert Evans, I forget his first name. They did the exact same thing as the guys who recreated the polio virus did, except they did it for horsepox, which is a reconstruction, which is sort of a cousin of smallpox, except it infects horses, as the name would suggest. And this took them 6 months with a small team of researchers and about $100,000. They just brought back this dead form of horsepox, which had been eradicated since the 80s. And they used this to sort of create a vaccine, right? It was a sort of test to see that it could be done. And they used it for a good reason, obviously. But the question remains, if you can synthesize horsepox, you can synthesize smallpox. They're not all that different. So that's also a thing that you can do, that you don't have to just create a new organism. You can just, buy parts of it. And everybody's heard of CRISPR-Cas9. The CRISPR-Cas9 system can essentially cut DNA wherever you want it to, and this creates double-stranded breaks, and then you can introduce new DNA into that break, and you can just sort of splice together genes. So you can take virulence factors, theoretically, from one very virulent bacteria that doesn't really transmit from person to person, and you can put it into the genome of a bacteria that does transmit from person to person. You can sort of take out the genes that code for the things that antibiotics destroy in bacteria. You can create something that's antibiotic resistant to multiple antibiotics or all antibiotics. You can just pick and choose what traits you want these things to have. And so this would raise the mortality rate, and you can also use the CRISPR-Cas9 system to create retroviral function, which is like the integration of a virus genome into a host chromosome. So, for example, AIDS or HIV is a retrovirus, and that's why it's so hard to combat, because it's literally integrated into your genome of the cells that it infects. So yeah, these are some problems that we can run into with with the advent of modern microbiology. And there's been a lot of call to regulate these sort of things, regulate who can use CRISPR-Cas9, who can synthesize oligonucleotides, there's been talk of a database that could be created to compare every sort of ordered oligonucleotide to a range of already known pathogens, genomes. But there's a bunch of ethical questions there. These guys did this to create a vaccine and to prove that you could create a vaccine from things that, are not alive anymore. And thus you can start to create sort of treatments based on these things. And how do we regulate this if it's being used for, good purposes, for leaps in modern medicine? And can we? Because we would have to have the companies do this, and we would have to impose laws on these companies. And that sounds like a logistical nightmare for someone who's more versed in politics to talk about. So, knowing all of this, in the wake of a bioweaponry attack, how does the world end, so to speak? Here are, I sort of created a mock scenario. Here are some assumptions that I made. For example, I assumed a non-state cult because they would just sort of want to watch people die and they wouldn't really care about it getting out of hand. In this sort of hypothetical scenario, it's a multi-antibiotic resistant bacteria. Based on the model of Francisella tularensis that I showed you earlier. So the mortality rate would be the lower end of the spectrum for Francisella tularensis, so 30%. And so the infectivity will be sort of similar to the infectivity of smallpox and rubella. So highly infectious, but not the ridiculous sort of 90% of you will get infected of the measles. It would have a 3 to 5 day incubation period before the onset of symptoms. You would have death or recovery in 14 days. And this would be my model takes place in December in New York City based on the World Health Organization's projections of a Francisella tularensis aerosolized attack, which said that in a city of 5 million people, there would, with a 50 kilogram aerosolized weapons, about 250,000 people would be infected. And I sort of had it in my model that it would spread over a range of travel hubs, so it would spread to the world as fast as possible. So, for example, JFK Airport, which has about something along the lines of 1.2 million people going in and out of it every day. And so what I based this on was an SIR model. SIR is a sort of equation that we use to model the spread of disease. It's a bunch of math. I'm not a math person, so I just put it into R and let R do the work for me. So you have your S, you're susceptible, so people who haven't been infected, your I, the people who have, and the R, the people who have recovered. And out of the people who have recovered, so to speak, these are the people who have moved from the infected stage to the non-infected stage. So in our model, 30% of these people, give or take, would die from this and not actually recover. So in week 1, people would be mostly not really noticing that this happens. It has a 3 to 5 day incubation period. These 250,000 people who are infected by this would just sort of go about their daily lives. And about sort of midweek, they would start seeing symptoms. And when you start seeing symptoms, even if it's like flu season, you don't always go to the doctor right away. You might go to the doctor if it persists for a long time or if it gets really bad. So we're going to say, at the end of the week, a whole bunch of these people just start going to the doctor and start, reporting these symptoms. Because it's December, in the height of flu season, a lot of these would be dismissed as cases of the flu. And because no one's dying yet, people would just be sent home with a lot of like medication for that. So this would be the infected count at the end of week 1, assuming that every single one of these people are around other people and are the model is based on a homogenous society that like mixes with a bunch of people. So it's a very simple model, not necessarily indicative of reality. And so I'm going to sort of divert from that model later on, but assuming that no one's caught on yet, this is the approximate infected count at the end of week 1. And so in the start of week 2, doctors and people would start realizing that something's up. The flu is really bad this year, or something along those lines. The World Health Organization probably would wise up to the sort of unrealistic nature of this pathogen at this time, and they would start sort of doing research into it, and they start seeing you would start seeing international labs popping up to sort of study this. And by the end of day 14, 75,000 people will have died from those original 250,000. And that's assuming that you die at the 14-day mark and not before. So people would start panicking at this point. A lot of people would probably start moving out of, densely populated city areas and into rural areas, where they would then infect these susceptible rural communities. So at the end of this 2-week period, 13 million people approximately will be infected in total. And of these 13 million people, 250,000 are already going to be dead, and 3 million will die from the currently infected population. So a little more statistics for you. There are approximately 25,000 registered law enforcement officials in LA, yet there are an order of magnitude or more members of known gangs in the same area. And so what happens when people start panicking and people start, wanting to hole up in their homes with their families and sort of wait for this to pass? We're going to see worker absenteeism. And this goes into, the police population. And so you're going to see a high rise of crimes as people start panicking and people start looting around week 3. And I modeled this sort of after the looting and the rioting that takes place in the wake of natural disasters, because obviously we haven't had sort of a pandemic on this scale in modern society. Around week 3, you would have hospitals running at about 50% manpower, and that's data taken directly from the SARS outbreak in Hong Kong. And so you'd have a massive sort of shortage in healthcare and people who know what to do. And you would also have a massive uptick in crime. And depending on what the political system is like at the time, you could have the institution of martial law in certain countries and quarantines of buildings and cities that are infected, because at this point, a bunch of people are dying, somewhere on the order of like millions worldwide. And here's where I sort of divert from my model, because there's too many factors that go into this that you can't just plug into a mathematical model, because humans are unpredictable, and that's how we work. So the current state of the world is that you have military soldiers just going everywhere trying to enforce the law. But what they're actually doing is they're continuing to introduce this pathogen to people who aren't sick in the military, and then these military people get pulled back and sent somewhere else, and pulled back and sent somewhere else. And if this disease works, as Francisella tularensis works, where it can transmit between humans and animals, it is likely that you could theoretically transmit this disease to the animal population and keep it there for a while. So even if you stayed in your basement for weeks and somehow had enough food to sustain yourself and sort of emerge later, you could still get sick if the animal population was infected in your area. And so in week 4, we would see sort of like absenteeism levels rise to a new level that we've never seen before. And I was even if you assume that the grid operators in the United States are, the most patriotic people ever and are so dedicated to their job that they will keep working even though, there's this massive pandemic going on, even they, at this point, with the violence that's going on around them and the disease that's ravaging the entire world, they would probably not want to go to work and, go through these public areas and risk getting infected. Or maybe they are infected themselves. And our grid is actually very, very fragile, and it needs these grid operators to direct power to different places and to prevent failure. And so what's likely to happen is that if enough people if enough grid operators leave their posts, and obviously I don't have a concrete number because no one's doing research on how many grid operators need to leave their posts for the grid to fail, we would see large, widespread power outages. And that holds a lot of problems for basically every sort of aspect of modern day society. Communication, the cell towers would stay up for a little bit, but how long are they going to stay up with no one to man them? And how long are they going to stay up with everybody trying to call everybody else and all of their members of their family to see if they're okay? The sewage system would probably suffer failure, and you would have these streets that are very dirty, and you would get a lot of secondary infections. And at that point, it's safe to say that even if the world doesn't end, and even if we don't kill the continent, modern society as we know it grinds to a halt. I actually read an entire book to research what happens after like a massive power outage, because nobody actually knows. And basically what they said is we have nothing really in place to fix this and no real protocols to help people from this. So that's essentially the stopping point for us as a civilization. So that is how to kill a continent, theoretically.